Hello, everybody, and welcome back. Hopefully, y'all had some success with the coding challenge problem. Again, just as a reminder, if you are having any issues or struggling, yeah, please post them your questions in the Slack, and the TAs will be happy to help you. Also, I'm going to go ahead and walk you through this exercise and see what happens. Let's see. Share my screen. Great. So y'all should be able to see my notebook. Again, here's the challenge problem. Now, before we get going on this, there's actually a little addendum down here that talks about the trade-off value. I mentioned this previously, but the trade-off value is actually very important when we are considering these like simulation results. Again, I mentioned that we chose a trade-off value that was previously validated, and I showed you some examples where you could see how changing the trade-off or choosing not to use the trade-off could result in vastly different results. With regard to our data, we might be interested to know, how varying the trade-off will affect the results. So while we work on the challenge problem, I'm gonna get this going so that we can take a look and see what the results are going to be for varying the trade-off. Here's just an example of what that result's gonna look like. We'll be able to look at the fraction of taxa growing as a function of the trade-off. And you can see in this particular example, if you want to capture, say, 80 to 90% of the taxa, the trade-off is somewhere maybe between 0.6 and 0.8. But again, we'll see what that looks like for our data. Now, let's come back to this challenge problem and see if we can figure out how to generate this heat map of C. diff imports. So the way I like to do this is I generally try to figure out one piece at a time. So I'm going to go ahead and make a new coding block down below just so we can kind of test things as we go. So the first thing we want to do is get the exchanges. So to do that, we'll say exchanges, exchanges equals, you can get this from the growth data frame dot exchanges. And actually, I spelled this wrong. So now, if we want to look at this variable, we'll do it like that. So we can see in this first step how things are looking. And oh, yeah, apologies. I realize this isn't going to work while this is running. So let's go ahead and just stop this so we can focus on this for a moment. We have to, nope, don't want to do that. Okay, so going back here. So yeah, you can see this is how you get the exchanges. Great, first step done. So I'm gonna copy this and put it right here. Okay, next we want to get the C. diff exchanges. So let's go ahead, and the way we're going to do that is pretty similar to how we were able to sort of filter things in some of the previous steps. You can do this. This is an approach that's really common to the pandas data frames that we're working with. So to select anything in the data frame, you can, or to filter anything in a data frame, you can first apply some kind of logic. So in this case, I'm going to use the taxon to filter for Clostridioides. And I know I'm probably going to spell it wrong, so I'm going to go ahead and find it right up here. And then we'll come back down. And I'll add this. So let's see what this does. I'm saying, let's look and see let's, for every taxon in our data frame, let's compare it to the string Clostridioides and see if they're equal. So if I run this, what you see is that it generates a true or false table. Now, if we put that back inside the data frame, so we're feeding the true or false table into our data frame as such, then this will allow us to select the exchanges that are specific to Clostridioides. So you see, we now have a data frame where you have all the exchanges specific to the taxon Clostridioides. So that's great. Second step, done. So back here. I'll go ahead and copy this so that we can keep our testing framework here going. Great. So now we want the C. diff imports. So to get the C. diff imports, we can do essentially the same thing. Equals changes. Again, we're going to use this ability to select. And one thing that's really important to notice is this direction column in the exchanges data frame. You can see it says import or export, and that's associated with the sign. So if the flux is negative, it's an import. If the flux is positive, it's an export. So if we want to look at the imports, we'll just do pretty much the same thing that we just did. I'll do exchanges, oops, dot direction equals import. And now if I do C. diff imports, look at that, we got it. So now the data frame has been filtered down to the features that are just the imports. So I'll go ahead and copy this and do that here. Now you see the next thing we want to do is add the metadata. So we've done that actually many times now. So you know, or you should be familiar maybe with the pd.merge function. So I'll do cdiff imports and metadata. And then the thing we're going to merge on is going to be the sample ID. That way you can make sure that each sample is getting the correct metadata. So you can see if I run this, it does just what we were expecting to do. You have the imports, and now you have all the metadata. So we're doing pretty good so far. So I'll put this here. And I'll copy this just so we can keep doing this one line at a time and see what our results are looking like. Next, we are wanting to do the pivot table. Now, the reason we're doing the pivot table is because we want to make a heat map that's sort of like a grid where each row is a metabolite and each column is a sample. And if we are looking at this imports data frame, we see that's not the structure that it's in. You see it's sort of like a long format where each row is a sample, and then you have, for each row, you also have a different metabolite and flux, etc. So to do the pivot, it'll be exactly as it was up above. So I'll go ahead and just scroll up here and copy this. So paste it here. Now importantly, we're working with a different variable here. So we're going to do this. And then the thing that's going to be a little bit different is the column is no longer called name, it's called flux. Or actually, sorry, it's reaction. and the values are the fluxes. Great. So now you can see we have a square or rectangular table. And I kind of mentioned this before, but not every sample will necessarily have an associated flux with it. So that's why you get all these NANs. And that's just because, yeah, they weren't able to compute, or they just didn't show up for whatever reason. They weren't relevant in that sample. And so we're going to want to do this step. Which fills all the NANs with zeros. Great. We're doing pretty good so far. So I'm going to get rid of this comment. That's fine. So C diff imports, put this here. Great. Now, this is sort of an optional thing, but so this could work without this step, but it would be nice to have the reactions in a more human readable format. You see right now, it's sort of an abbreviation. The EX is telling you that it's an exchange reaction, and then they have sort of like an abbreviation for the different metabolites. So if we want to get that into a more human readable format, first we're going to want to import the annotations, which is what's noted here. So we'll say annot equals growth. Again, going back to our original table of information, and you can see dots, and it usually gives you a bunch of options. So we want the annotations option. Now, if we look at that, you see we get a table of annotations where the index is the reaction. That's great because the index for our table is also reaction, so there should be a pretty easy way to map them. But let's see, yeah, let's just confirm. Yep, index is already reaction, so we don't need to, yeah, you can see that that was true here. I think I did it up here just to be safe in case it wasn't, but it looks like that's not necessary. So now, if we want to map the annotations to our table, then what we'll want to do is create a dictionary. So first, let's find the column we're interested in. Name is the column that we're interested in. You see it has a more human readable format. So for instance, this metabolite or exchange reaction, EX underscore 20 or 20 BUT is 2-oxobutanoate. So if we want that information, we'll go to that column. So annot.name. One thing that's worth noting is with pandas, you can use the dot and then the name of the column, like dot name, or you could use the square bracket and do it like this. Both are equivalent and will work in most cases as long as there's no spaces in the name. So I could do it like that. Now we want to make a dictionary. So I'll do it like that. Add the 2_dict function. And now you see what that does is it creates a dictionary where you have the key of the dictionary is the abbreviation, and then the value associated with it is the thing that we're interested in. That's great. So now if we want to map our index using this dictionary, we can do so as following. So we will get our data frame of interest, C diff imports. We'll look at the index. And we'll do dot map. So this is going to take the index values and then map them to using this corresponding dictionary. So if we run that, you can see the index gets converted, or like the result of the mapping is converting from those IDs to a more human readable format. So let's go ahead and copy some of these things and move them up to our code cell here. So that's that. And then that next step is here. Great, so we're all done here. So if I run this, oh, I missed a step. Let's see, oh, yep, I forgot this guy. Let's just delete that. No, we actually do need to do that. So let's copy this. And also I see I made a mistake here. So let's get rid of that. Yes, that is actually one step I forgot. We do want to normalize our data as we did previously. I mentioned that we are using a log normalization because the fluxes vary on an exponential scale. So if we go up here, we can sort of copy this little block of code that applies the normalization. So if we paste it here, we can replace exchanges with cdiff imports. And just to clarify what's happening here, so we're doing this apply function. So it's applying the operation that's inside of the apply function to the values in the data frame. And you can see here there's an access argument that tells you to do it whether row wise or column wise. In our case, that doesn't really matter. And you can see the thing that it's applying is this lambda function, where it's saying take the value, whatever it is, which will be the individual flux units, and apply a log transform to it. Let's see, and then let's just double check. This might give a issue here. Let's confirm. Okay, annotations. Yeah, that looks good. I think we might need to apply an absolute value here because we have some negative values. Oh, and then also a slight error here. Let's see. Yep, let's get rid of that. Great. So now you can see the table has been transformed. We have in the format that we like, where the rows are the different metabolites and the columns are the different samples, and they have all been log transformed. Great. So now we can make the heat map. To do that, we'll use the same little code block that we used up above. And we can just get rid of all of this. In fact, I'll just get rid of that altogether. Paste that here. And then we will want to just plug in our cdiff imports here. I think, yeah, there's actually one more step here that I missed. If we go back up here, so I applied the log transformation, but if you remember up above, one of the things that we did was we sorted the data frame by the variance so that we could look at the things that were most different between the samples. So we'll want to do that again. Imports equals cdiff inputs dot re-index. So we're re-indexing the data frame, it's changing the orientation. And then we're going to do that using the variance, which you calculate with this var function. And then we're calculating it across the samples. So we'll use the axis equals 1. So it's saying calculate the variance across the rows. And then we're gonna, once we calculate the variance, we wanna sort those values. And we want it to be from largest to smallest. So we'll give it this argument ascending from this false. And then ultimately we want the index. So that should sort everything. And you can see already it seems to work. Like our top most variable flux is for this lactate, which you can see appears to be high in the pre-FMT for the most part and low in the healthy samples. Okay, so now we should be able to just make this heat map using the top 50. And one thing to know is your results might look slightly different than mine. That's because every time we run this operation where we're estimating the growth rates and solving for the fluxes, it's possible to get slightly different solutions that are equivalent to each other. It's akin to that issue that I mentioned before with FBA, where there are maybe potentially multiple viable solutions in the solution space. We use cooperative trade-off flux balance analysis to sort of limit that. And in addition, with the medium, the constraints, the imports, and the parsimonious flux balance analysis that minimizes the enzyme burden. But even with all those constraints, you can still have multiple viable solutions. And so, yeah, here are the results. And in this particular case, we see that we get a nice separation between our pre-FMT and our healthy samples. And you can see if you look at some of the metabolites that are different, you see, again, the lactate. There's also some amino acids like leucine and isoleucine that appear to be different. Mostly it looks like amino acids and sugars are the things that are differentiating the niche of, or the metabolic strategy used by C. diff in across these conditions. So that's great. We can see that, yeah, C. diff is consuming different things in our samples. And that difference in its consumption patterns could be associated with the consumption patterns of the community. And it's also likely associated with the difference in growth rate. So we focused on consumption rates here, but another option would be to look at the production rates. So for instance, we might be interested in the production rates of the community and see, is the community maybe producing something that C. diff is consuming? So if we wanted to do that, we could actually copy this whole code cell here. And then I'm just going to put it right below this heat map. And pretty much there's only going to be a few things that change here. So we're going to import the function production rates. We don't need to import that again. And then we will change that to that. So we're now calculating the production rate. What are the things that are produced? And then everything else will likely be able to stay the same. Okay, so once again, we're calculating, producing this table. Now it's production fluxes in the community in the absence of C. diff. And if we wanted to visualize that, we can go ahead and make another heat map. And look at that. It not only is the community consuming different things, it looks like the community is also producing different things. And yeah, if we look at some of the things that are produced, we noticed earlier there was a difference in some of the amino acid and sugar metabolites for C. diff in terms of what it's consuming. And one of those metabolites we noticed was isoleucine, and it looks like it's being produced in the pre-FMT samples and not so much in the healthy samples. If we scroll back down to our C. diff heat map, let's see if that matches up with our expectation. So isoleucine is consumed in the pre-FMT, but not in the healthy. And it is produced in the pre-FMT and not in the healthy. So yeah, that's just showing that the community, somebody or collectively in the community is producing isoleucine, and that isoleucine is being consumed by C. diff, which could be helping it, is very likely helping it engraft. And so if you wanted to, you could go through and try to identify additional exchanges that might be supporting C. diff. Similarly, you could imagine, you could look at the consumption fluxes and see if there are certain things that are maybe that are competitive. So for example, let's see, looks like acetate is something that's consumed in the let's actually look for something that's consumed in the healthy, but not in the pre-FMT. So let's see, there's quite a few here. Maybe let's look at guanosine, just as an arbitrary one, or glucose. So both of those are consumed in the pre-FMT, but not the healthy, or sorry, they're consumed in the healthy samples, but not the pre-FMT. Let's see if that matches up at all with our C. diff consumption. So let's look for glucose. Let's see, where's glucose? Here's glucose, okay. And it looks like it's also consumed in the healthy, but not the pre-FMT. So there might be some competition happening here in the healthy samples, since both C. diff and other genera are both consuming glucose. And I believe the other one we were looking at was glycolate. Let's see. That one, I might be just missing it, but I do not see it here. So it might not be something that C. diff cares about. Anyways, this is just one way that we can sort of use this analysis of community and individual fluxes to be able to sort of deconvolute what might be happening in these communities. Okay, so now I'll go ahead and try to run this again. And while this is running, I will use this time to kind of show you some other little tidbits. So if you're interested in using MyCom, if you'd like, you can kind of use the framework that we've set up for you here and run it in Google Colab. One thing I mentioned, though, is that there are some limitations here, like the resources available to us on Google Colab are somewhat limited. And so we have to, we're sort of restricted by our processing power. And also, ideally, you'd want to have a more powerful solver that is better at solving these types of problems. We're currently using a free kind of open source one that's not, it obviously works, but is not ideal for these types of problems. So if you're interested in setting up MyCom like on a server or your personal computer or something, or if you just want to learn more about MyCom, you can actually go to the GitHub page, MyCom Dev here. And here you can find a bunch of information, much more information about, for instance, here's the MyCom Python package. You can, there's a lot of kind of additional information you can read up about with regard to, the Gibbons Lab or ISB or, some of these other institutes where the development took place. You can see there's some information, like for instance, installing it's fairly easy. You can access documentation here or use these forums to get help if you're having problems. Additionally, if you go back here, there's some information about the QIIME 2 interface, sort of the same sort of thing. This has some information about setting up Cplex, which is one of the better solvers that's available. My understanding is this is You can usually get Cplex with an academic license, but otherwise, if it's for commercial purposes, it can be quite expensive. But this, if you use this solver, it actually can allow you to get much more accurate results. And there's some other information here to set up your QIIME2 interface you can read up on. And then finally, there are things like different databases that can be considered. We used the Agora 1 database. There's more recently been an Agora 2 database that came out that is expanded on the number of models that are available. So that's something you can consider. You can also use CarvMe or other databases that are compatible. And then finally, today for all the analysis that we ran, we used this average European diet, which is sort of just an assumption that we made to make things simple. But that is obviously not relevant in every context. For instance, you could go back and see last year's microbiome course where diet was a big focus of that course, and trying to do these types of simulations for indigenous folks and folks that are not typically represented in microbiome data. And in those contexts, the diet is very relevant as an average European diet that can't be applied to everybody, or shouldn't be. And so to that point, if you're interested in using different types of media or different diets, you can come to this GitHub page and look at some of the available media. Here's this table. So the Austrian average diet is the one that we've been using as the average European diet. There's also high fat, high protein, low carb diet, high fiber diet, a fermented foods diet, as well as baby formula. So these are just a few that are already available. You can also try to formulate your own diet if you'd like, using these as sort of like a framework for that. And actually, to that point, a great resource that a lot of this is sort of based upon is the virtual metabolic human, which you can find here at vmh.life. And this can be a tool that would allow you to potentially build your own custom diets if you're interested, or if you want to kind of investigate some of the metabolic models that are available. So if we could start here, we can, for instance, look at gut microbiota. And these just kind of give you a list of all the different models that are available. You can kind of like look at more if you'd like and get some additional information. For instance, the genes or some of the carbon sources in this case, or fermentation products. You can see this guy seems to make acetic acid and butyric acid, so it's a butyrate producer. Those are things, so you can learn more about some of the models. You can also sort of like look into more nutritional information. And look here, they're telling us that they've made some updates. So for instance, they have different diets here that have been put together. And so you could use this portal to kind of look more into diets and look up specific foods and the fluxes associated with them, so you could build your own diet. Anyway, these are just some of the resources that are available, and I encourage you to investigate them further. All right, so if we pop back over to the slides just for one moment, the last thing I'd like to mention is just, we've made it to the end, and this was a big team effort. So we just like to thank everyone who contributed. If you were here for yesterday's course, Nick Bowman taught that. And of course, there's so many others, Sean Gibbons, who this all originated from, as well as all our TAs and speakers and all the people that helped keep this running in the background. So thank you, everybody. And stick around for tomorrow, that we'll have a symposium with quite a few speakers talking more about some of the things we've touched on these past couple of days. And with that, we're all done.
Say goodbye, but there are a couple of questions that came up, so maybe just do a quick talk about that. So one was
Yeah, this,
about the scale of that, what it was telling us. We can talk about that.
okay. Yeah, so we got a few questions. So yeah, let's address some of those. You mentioned the scale of the heat map.
Yeah, they were asking about color map scale. I don't know.
Yeah, so here, if we look at the heat map, you see this color bar on the side. It goes from -6 to 2, dark colors. So this is a log scale. So negative numbers are very low. So where the log value is -6, that basically -6 and anything below that, even something as low as -4 is pretty close to 0. So that's telling you dark colors are very low flux. And then colors that are kind of more in the green, yellow spectrum are high flux.
Great. And then one final, I guess, is we use 6 units today, but can you also use metabolics for visualizing?
That's a great question. Yeah, the question was regarding use of metagenomics. Yeah, so today we use 16S because that's what was available, and also because it allowed us to kind of easily construct these models in a reasonable amount of time and do these simulations. But if we wanted to, we could actually use metagenomics. That's actually how MyCom was originally formulated, to leverage metagenomics to better map individual taxa to, say, the species or maybe even the strain level, depending on the resolution of your metagenomic data. So yes, you can definitely use metagenomics with MyCom. I would encourage you to look at the documentation for that, or even the original MyCom publication, where that is all sort of explained. Great. All right, so yeah, before we go, we're going to hear a few words from Christian Diener. And yeah, thanks again, everybody.